Good morning. It's April 10th, and this is your daily brief in science. Here's everything you need to know. A major breakthrough in neuroscience has been achieved with the mapping of approximately 84,000 neurons within a cubic millimeter of mouse brain tissue. This groundbreaking project, involving over 150 researchers, unveiled around 524 million synapses and 5.4 kilometers of neural wiring, marking a significant step forward in our understanding of brain connectivity. The research team employed advanced microscopy, artificial intelligence, and 3D reconstruction techniques to achieve an unprecedented level of detail in mapping these neural connections. They studied a genetically engineered mouse with glowing neurons to analyze how its visual cortex reacted to various video clips, including iconic scenes from The Matrix. These findings, published in a collection of 10 studies in the journal Nature, represent a significant advancement in neuroscience, comparable to the Human Genome Project. The comprehensive dataset is publicly accessible and aims to enhance our understanding of brain function and disorders like Alzheimer's and autism. The project revealed new principles of brain organization, notably that inhibitory neurons coordinate activity selectively rather than merely suppressing it. Co-leader Sebastian Seung noted that this foundational work will assist in identifying abnormal connectivity patterns in brain disorders, such as autism and schizophrenia. Spanning 9 years, the project produced the largest brain circuit map to date, essential for understanding brain function and diseases. Although this mouse connectomy represents just a fraction of the entire mouse brain, it sets the stage for future studies, including the ambitious goal of mapping the human connectomy. This unprecedented combination of functional and structural mapping highlights a significant achievement in the field of connectomics, showcasing the complexity of neuronal connections. Funded by the U.S. Office of the Director of National Intelligence and the National Institutes of Health, this collaborative effort underscores the importance of interdisciplinary cooperation in advancing scientific research. A recent study published in JAMA Neurology has found promising evidence that GLP-1 drugs, such as Ozempic, may lower the risk of dementia in individuals with type 2 diabetes. This research involved a meta-analysis of 23 randomized trials, encompassing over 150,000 participants, and indicates potential protective effects of these medications on brain health. Data for the study was collected from more than 92,000 individuals aged 50 and older, with type 2 diabetes across several states, including Florida, Georgia, and Alabama. The findings are particularly significant in light of the fact that there is currently no cure for Alzheimer's disease, which affects approximately 6.7 million Americans aged 65 and older. Experts, including Professor Martin O'Donnell, have pointed out the public health implications of the increasing rates of both diabetes and dementia. Longevity expert William Kapp noted that metabolic issues associated with diabetes could also have a detrimental effect on brain health, further supporting the study's conclusions. However, caution is warranted. Professor Tara Spires-Jones emphasized that while the results are encouraging, the drugs come with side effects and do not guarantee dementia prevention. Additionally, a concurrent meta-analysis raised questions about the effectiveness of SGLT2 inhibitors in reducing dementia risk, highlighting the necessity for more long-term data. Ongoing clinical trials are currently investigating whether semaglutide can protect against dementia in individuals without diabetes, aiming to clarify its broader implications. Experts are advocating for more prospective trials to strengthen the evidence regarding the role of GLP-1 receptor agonists in reducing dementia risk, especially in non-diabetics. With 1 million individuals diagnosed with dementia in the UK, effective preventative treatments are urgently needed given the lack of available options. A new 3D scan of the Titanic has emerged, revealing critical insights into the ship's tragic sinking. This scan, executed by underwater robots that descended 3,800 meters to the ocean floor, is composed of over 700,000 images, creating a detailed digital replica of the wreck. Among the significant findings is evidence that engineers worked tirelessly to keep the lights on until the end, which facilitated the safe launching of lifeboats during the chaos following the iceberg collision, ultimately saving many lives. Advanced simulations from the scan indicate that the Titanic sustained multiple small perforations in its hull, each roughly the size of an A4 sheet. These small breaches collectively allowed water to flood several compartments, leading to the disaster, as the ship was designed to remain afloat with only 4 compartments flooded. Additionally, a previously unreported porthole likely smashed by the iceberg corroborates survivor accounts of ice entering passenger cabins during the collision. The 3D model illustrates the violent breakup of the ship and confirms eyewitness reports of engineers maintaining power until the end, highlighting their sacrifice. Captain Edward John Smith's final moments are also a focal point, with survivors recalling his calm demeanor and his last command urging passengers to evacuate. Witnesses noted Smith's bravery as he assisted women and children into lifeboats, with his last reported command being, "Be British." As the wreck deteriorates rapidly, projections suggest it may vanish within the next 40 years, emphasizing the urgency of ongoing research. This project, featured in the upcoming National Geographic documentary, Titanic: The Digital Resurrection, marks the first comprehensive mapping of the entire wreck. Despite these new findings, researchers like Park Stevenson emphasize that the Titanic still holds many secrets, and further exploration is essential to fully understand its story. A recent study has found that restricting eating to daytime hours may significantly benefit the heart health of shift workers. This research highlights the potential for timing food intake to reduce cardiovascular risks associated with night shifts. Professor Frank Shear, a lead researcher, pointed out that circadian misalignment increases cardiovascular risks. The study suggests that when individuals consume food at night, they exhibit heightened cardiovascular risk factors, whereas those who eat only during the day show stable health metrics. Conducted at Brigham and Women's Center for Clinical Investigation, the study involved 20 healthy young participants who were isolated from natural light and time cues for 2 weeks. Participants adhered to a controlled, constant routine protocol, which helped distinguish the effects of circadian rhythms from environmental influences. During the study, participants remained awake for 32 hours in a dim environment, consuming identical snacks hourly before being assigned to either daytime or nighttime eating schedules. Researchers evaluated various cardiovascular risk factors, including autonomic nervous system markers and blood pressure, before and after the simulated night work. The study's rigorous control over sleep, eating patterns, and environmental factors allowed for clear conclusions about the impact of food timing on health. However, its limitations include a small sample size and a short duration, which may not fully reflect the chronic effects of nighttime versus daytime eating. Previous research has established a connection between night shift work and significant cardiovascular health risks, underscoring the need to address circadian misalignment. Additionally, animal studies have suggested that aligning food intake with circadian rhythms can mitigate health risks linked to nighttime wakefulness, further supporting the findings of this human study. The results of this research, conducted by teams from the University of Southampton and Mass General Brigham, were published in the journal Nature Communications. Scientists at the Van Andel Institute have made a significant breakthrough in the battle against pancreatic cancer. They have developed a method for identifying and classifying subtypes of pancreatic cancer cells using unique sugar signatures known as glycans. This innovative technique, referred to as multiplexed glycan immunofluorescence, leverages advanced software and imaging technology to analyze the composition of cancer cells within tumors. The research team has refined their approach to detect glycans released into the bloodstream, making testing less invasive and more patient-friendly compared to traditional surgical methods. Blood tests for glycans are not only quicker but also more cost-effective, enhancing the overall patient experience. This advancement has the potential to lead to earlier and more accurate diagnoses of pancreatic cancer, which often presents no symptoms in its early stages. According to Dr. Brian Hobb, the study's corresponding author, this method will allow physicians to tailor treatments more effectively by revealing the specific subtypes of cancer cells present in a tumor. The study, co-authored by Braylon Binkowski and Zachary Klamer, is supported by a diverse group of collaborators from various institutions. The research team continues to validate this method and aims to discover glycan signatures for rarer cell types and other gastrointestinal cancers. They are also exploring the potential to apply this method to detect and characterize other gastrointestinal cancers. Pancreatic cancer is notoriously difficult to detect early, with only 15% of cases diagnosed in time for surgical intervention due to the lack of noticeable symptoms. While this method is not yet available in clinical labs, researchers are optimistic that it could become a standard diagnostic tool for pancreatic cancer within a few years. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.